Good afternoon, ladies and gentlemen.
Good
I
afternoon.
think now we are in the same wide range of time. So now we all have in the afternoon time, right? Okay. Now we are in the session 2. Is about regional advances in the Pacific. So in this session, we have 8 speakers. The first speaker is, before I introduce the speaker, let me introduce myself. Sorry. So my name is Nadiarti, mostly called as Nadia or Arti. I'm from Fisheries Department, and my research interest is about aquatic species biodiversity. And I'm accompanied by Ibu Elmi, Dr. Elmi Zainuddin. His research interest is about seaweed development and biotechnology. She is also from Fisheries Department of Hasanuddin University. Okay, all right. So we have 8 speakers here for this session. Let me introduce each of the speaker. The first speaker is Petrus Rani Pongmasa. He is a senior researcher at the Research Institute for Seaweed Culture in Gorontalo under the Ministry of Fisheries and Marine Affairs of Indonesia. He was formerly stationed at the Maros Research Station in South Sulawesi. At the moment, he leads a team conducting research on seaweed farming and domestication of new species across Indonesia. He has involved in many bilateral aquaculture and fisheries research partnerships between Indonesia and Australia. The next speaker is Rajuddin Syamsuddin. He is a professor in the Faculty of Marine Science and Fisheries, Universitas Hasanuddin, Indonesia. His PhD studies at the University of the Philippines in Los Banos were followed by postdoctoral studies and experiences with a university in France. His main interests focus on the eukymatoid seaweeds, with special interest including seaweed propagation methods, etc. The next speaker is Ma'ruf Kasim. He is a professor at the Faculty of Fisheries and Marine Sciences, Halu Oleo University, Kendari, Indonesia. His doctorate in marine ecology from Hokkaido University, Japan. Now he is active in international professional organization in the marine and fisheries and the marine conservation sectors. He also a guest lecturer or invited professor at Kyoto and Hokkaido universities. He is a team leader of 28 competitive national research projects with many national and international seaweed-related publications. He also an innovator with several patents and awards. He has a special interest in eukymatoid seaweed cultivation and ecology. Next speaker, Lideman, a researcher at the Takalar British Water Aquaculture Station of the Ministry of Marine. He is alumnus of Kagoshima University, Japan, and JSPS Invitational Fellowship for Research in 2019. He involved in Indonesia-Australia collaborations. He got patent for Brasileria species reproduction from spores in 2020. His research interests include the sexual and asexual propagation of seaweeds, seedling acclimation, the effect of environmental factors on white and fawn seaweeds, etc. The next speaker is Xiao Ting Hu. She's based at the Ocean University of China. Her research about seaweed includes nutrition analysis, functional components, carbohydrate, phenolic compounds, pigments, isolation, purification, biological activity, characterization, and also and etc. Her research application include nutraceuticals, functional food, functional cosmetics, enzymatic production of oligosaccharides from seaweed as functional food for human intestinal health. In agricultural application, including seaweed fertilizer using alginate to make field covering membranes to replace plastic sheets. The next speaker is Ahmad Sadeh. He is a director, Department of Fisheries in Sabah, Malaysia, working at the Sabah since 1989. His PhD in food biotechnology, University of Strathclyde, UK, 1998. He is involved in a wide spectrum of fisheries activities, including mariculture, research, policy, and development aspects. Sabah Department of Fisheries is tasked by the Malaysian government to develop seaweed aquaculture industries in Malaysia. Dr. Sadeh and team are working with local communities and private sector to expand seaweed aquaculture and are actively engaged in seaweed research and development partnerships to increase production and diversity seaweed application. The next speaker is Day-1, Chinese Academy of Science, China. Dr. Duan and his team have over 30 years experience on seaweed genetics and breeding, seaweed nursery and development, seaweed metabolism and bioactive substances, and have a valuable experience with euchema cultivation in the 1980s. And his extensive knowledge on current kerajinan production status and trends in China. I think Prof. Day-1, Professor Day-1. The next speaker is Pepijin Pan Or. I'm sorry if I'm mispronounce the name. Pepijin Pan Or is a scientist at Wageningen University and Research Center, Netherlands. He has a background in GIS and over 15 years of experience in crop growth modeling, rice, potatoes, and etc. He recently, Pepijin has started modeling the growth of seaweeds at local, Indonesia, Netherlands, and global scales. I think that's all the introduction for our speakers for this session. And now we start a discussion based on the question provided from the participants in the web forum. But we also invite participants to provide a question in Q&A menu if there is any more additional questions. So in this time, I invite Dr. Petrus Rani Pongmasak. Dr. Pongmasak, are you there?
Yes. I hear.
Good afternoon, Dr. Komasa.
Good afternoon. Good afternoon to
So because
Adiati.
we are in the same area in Indonesia, so yeah, nice to meet you, Dr. Komasa. In your video, you're talking about growth and product quality of Capaticus from Indonesia, right? So there is
Yeah.
several questions we pick from the forum. The first question, were the various cultivars distinguishable from each other based on molecular taxonomy techniques? What do you think, Dr. Komasa?
Thank you, Paden. Prof. Nadiarti. Paden, can you
Okay, I will repeat
again?
it. Were the various cultivars distinguishable from each other based on molecular taxonomy techniques?
Um, we take a sample from another place from Indonesia, and we do analysis by DNA sequencing and sequencing in laboratory. Yeah.
I mean, from all various cultivars, are they distinguishable or can be distinguished based on molecular taxonomy techniques?
Yeah, we depend of molecular and is yes, we with the sequences we can see in the our result for name of species. We have get some name of the species from sequences, and we can compare variation between species with gene bank data and the sequences result.
Okay, so the cultivars can be distinguished using molecular taxonomy techniques, right? Okay, the next question. Do you have any thoughts on why certain regions fare better than others in terms of seaweed growth and quality?
About the quality, we just measurement about the gel strength and capapicus, and we can see in our videos the compare with quality about quality keraginan and gel strength of capapicus. There is no correlation between viscosity between another quality. And about the criteria of quality, we just take about keraginan and gel strength in this study.
So, what is the reason, from your opinion or your thoughts, why certain regions have better seaweed growth and quality compared to the others regions, to the other regions? What do you think?
Our we think about for the reason because every place in like in Indonesia, different place we get product, quality product very different because the place get uh, uh, what about water quality depend of the water quality and what the uh, uh, the water quality and then uh, uh, I, I'm sorry. Uh, the quality depend of the water quality and then I think the uh, uh, decision differentiation in the wet season and dry season and some parameter to I'm sorry. I want my English is very.
It's okay, Prof. Dr. Masa. So do you mean the different quality of seaweed, the different quality and growth in between different area, maybe because of the weather, the environment condition is different, including the water quality,
Yeah.
something like that.
The quality of seaweed depend of the water quality and then how the how long we cultivation in the water and then the quality very different about the nutrient in the water and then what about light and then salinity and then other water quality depend on the water quality and main main about the quality about the season in season in the every year. Yeah.
Okay. So do you think, what about the seasonal growth differences in seaweed quality and growth rates between different sites? Is there any, when comparing between sites, do you think, what do you think, how to account for these differences? I mean, can you get it?
Yeah, in different size in Indonesia. About I have say different size give quality different quality in Indonesia. Yeah.
Maybe you can answer in Bahasa and then I will try to translate it to make your questions going through flow in well. So ada kalau pada dua lokasi yang berbeda pengaruh musim mempengaruhi pertumbuhan rumput laut. Nah, menurut Bapak, apakah bagaimana pengaruh musim itu mempengaruhi kedua kualitas rumput laut yang berbeda pada lokasi yang berbeda?
Ya, itu sangat berpengaruh Bu karena kedua musim ini mengandung kualitas perairan yang memberikan kualitas perairan yang berbeda. berbeda sehingga musimnya berbeda, musim hujan dan musim kering itu memberikan kualitas yang berbeda, dan kualitas yang berbeda ini disebabkan oleh kandungan yang nutrien di dalam perairan yang berbeda, di mana musim kemarau itu lebih rendah dibanding musim hujan.
Okay, thank you, Dr. Komasa. So the growth and quality of the seaweed, it depends or influenced by the seasonal, by the season, yeah. Because different season, dry season and rainy season will affect the nutritional condition, something like that. All right. Because we are limited by the time, thank you, Pak Dr. Komasa, for your excellent response. So now we move to the next speakers. Professor Rajuddin Samsudin, are you standby?
Yeah.
Are you there, Prof. Rajuddin?
Yes. Can you hear me? Can you hear?
Hello, Prof. Rajuddin.
Hello. Can you hear?
Prof. Rajuddin.
Yes.
Are you there?
Yes.
Can you hear my voice, Prof?
Can you
Hello?
hear also my voice?
A little bit. It's not so clear.
Not so clear. Can you hear my voice?
Hello?
Hello. Hello. Can you hear
Maybe
me?
your microphone?
Should I there? Can you hear? Can you hear my voice?
Seems that you're far away from the microphone.
Can you hear? Can you hear my voice?
Maybe you should remove the headset. Try to
Can you hear? Can you hear my voice?
remove the headset from your laptop. Try to remove the connection.
Can you hear?
Coba dicabut Pak speakernya, anunya headsetnya dari laptop.
Ya, ya, ya. Bisa dengar?
It's not really clear.
Can you hear?
But
Can you hear my voice? Can you hear?
maybe
Can you hear?
we give your time to fix your microphone, and now I'm trying to move to the other speaker, and then we'll be back to you after this. Okay, Prof? Because we are running out of time. I'm sorry for this. Yeah. So I will. Okay. So for the next speaker, Dr. Ma'ruf Kasim, I will invite my partner, facilitator, Ibu Elmi, to guide the discussion. Ibu Elmi. Halo Ibu Elmi.
Yeah, okay. Thank you. Thank you, Doctor. Sorry. So, okay, thank you, Dr. Nadiarti. Yeah. And then for the next, I would invite Professor Dr. Ma'ruf Kasim.
Yes, I'm here. Yes, I'm here. Can you hear my voice?
Oh, yeah, okay.
Okay. I hope.
But also, I'm not looking your face. Not yet looking your face.
It's not clear. It's not clear. Okay. So.
Okay. Thank you. So yeah, Dr. Ma'ruf Kasim. Yeah. I have some question for you from the website. Maybe you have also read it. And yeah, the first question that I choose in here is, what are the constraints, yeah, associated because yeah, I want to explain first. You make research to compare the cage method with the conventional method, yeah, in turn for production and protect from predator. Just like that.
Yeah.
Okay.
I
And
compare
the question is, what are the constraints, yeah, associated with the cage method, and what is the acceptability rate of the cage method by seaweed farmer, and also did you have make a cost benefit analysis to see that this method is acceptable for the farmer compared to the conventional method, long line method? Please.
Yeah, actually, we do several research to compare between long line with the cage method, because we starting the research in 2019, and we tried to make comparisons of this, and we found that this is really different, because the specific growth rate is also different. While I'm presenting my result of my presentations. And actually, you know that this method we have for variants, and while we try to compare in between variant also there are different. And if we compare totally with the 4 variant with with between the 4 variant and the traditional one, it was really different. And even though the not so really different with the total growth, but the specific growth rate we found in during our research, they are different. And you know, that is actually I mean, that is we also analyze the cost benefit analysis of this. My student done this research, and then we found that the what it is, the in average of the floating cage is they have what is we call it beneficially high than the long line one. So that's why we really what recommended these innovations. Actually, between area there are different of the attach of the I mean, that is the different. I mean, different different what is this seaweed attachment by by fish or by sea turtle, but this is really different. That is what I'm.
I mean that there are constraints. It means there is a problem during use this method.
Actually, only few problem that we found in field, but because some people, some community is really want to use another method, another different method, because if they use the new method, then they will using the, I mean, that they will I mean, that they will need more money for infestation. So that's why they are still using the long line with low infestation. I mean that so that's why even not so much people in Indonesia, particularly in Southeast Sulawesi, but several people, several community starting to use these innovations.
Okay, I have the next questions. What about the economic balance? Because there's reduction of the loss of seaweed is interfere with productivity of the crop. And how about the react or the response of the to the epiphyte and the grazer is still the same epiphyte dominance in this method, or also with the grazer? How about that?
It's different. It's different. You found that the epiphyte, it's much more like to prefer to attach in the line and then the net and the pipe compared than the talus of the seaweed. From our research, we found that the variations of the epiphyte, macro epiphyte, attached on the talus of the seaweed is really low compared than attached in the long line one, because in long line one they have no protections. They are direct attached on the seaweed, on the talus of the seaweed. But if you're using the net, aside the to prevent the seaweed from the fish, also the several macroalgae is not really much attached on the seaweed talus.
And how about the grazer? The grazer is still,
the grazer also, yeah, of course. The first mind of these innovations is to protect the seaweed from the grazer, fish and also sea turtle, because several area like in Entebbe and Gorontalo, they have a lot of sea turtle, but in several area they have siganid one. So this is really prevent the seaweed from siganid, or also from the sea turtle. Like in Bali, Gorontalo, Entebbe, NTT, they are afraid to using it, and then they send me information related the functions of this, and then it significantly increase the productions. The new information that I found in Batam, that is, I allow them to using my innovations that they are used. It's really significant in protecting the seaweed from the fish and siganid.
It means this method has been practiced, yeah, by many farmers in the some area.
Yeah,
also
yeah.
in Sulawesi, yeah, in Sulawesi
Yeah,
also.
Yeah.
Okay.
Actually, Bu, this is variant. They have 4 variant. This 4 variant is they have specific using. The horizontal net is for the area is not so high tide or speed current. The vertical one is the several area with the not really high tide. But the long one, the long net, is for the what is this with for area is the high tide. So that's why this is even though they have 4 variant, but this is really I mean, this is really particular different in several area.
Oh yeah. Okay. Thank you very much. Nice explained from you, and we have many information about this novel method. Now we
Thank you very much.
go to the next panelist, Dr. Liedemann. I invite Dr. Liedemann for the next.
Yes, Bu. Well, me.
Okay. Yeah. So, Dr. Liedemann, yeah, I think I have also some questions about the after look the results of your research, which you observe about the effect of higher temperature and higher light intensity. Can you hear Can you hear me?
Yes, I can hear you.
Oh, okay. So, after look your result of your research, which you observed about the effect of higher temperature and higher light intensity to the eutrophic farming. Yeah, some question is want to you explain or you give the advice or recommendation about your result of your research to the seaweed farmer. it is acceptable, or it is easy to applicate it in for the seaweed farmer. Your recommendation or your advice for that?
Yes. From my result, I think the farmer can culture at the different site or different location uh, at different season. For example, in the we have a Banteng Regency. At that area, some of the farmer have several area. They have close to the beach and also far from the beach. So they can culture both of this area, but in the extreme condition, extreme weather, the farmer, for example, when we have only two climate, two season, dry and we call dry and raining season. For example, in the raining season, they culture it in the offshore. I mean, far from beach. But uh, in the raining season and not in the dry season, they culture little bit close to the beach because in that area there are several river. That's why from this result we can apply that in the dry season we can avoid we can culture in the not so high uh, not I mean not higher light intensity and also temperature.
Yeah, it's meant because the result of when in the dry season is the temperature is higher, yeah, than the rainy season, and you because one questions about that. Can you mitigate the impact in shallow water? Yeah, the mitigate the impact in shallow water by moving the techniques into deep water. It is yeah. What are the technical challenge if so?
Yeah, I think this is a good idea because in the deeper water we can culture. I mean, more deep in the deeper area, right? So some area, for example, in the Bali Island, there is upwelling. So at that area, the temperature is lower than another area. So we can culture Capapicus alvarezi better at that area. I mean, if we can find the area that has upwelling, the temperature should be lower than area that there's no upwelling.
Yeah, it's mean the
But
yeah, because
also there is the problem. If we culture in the shallow water, most of the Capapicus, I mean, there are there are so much mud, sometimes attached to the not only mud, sometimes the epiphyte attached to our Capapicus when we culture in the deeper area, but in the shallow water. Not in deeper area, the culture in the deeper in the deeper water column and in the shallow water.
And one question, last question. Does higher temperature and light intensity cause bleaching of the talus?
Yeah, of course. We have experience when we culture at the temperature 34 and light intensity 100. The Capapicus will bleach for approximately 1 week. But in the lower light intensity, it's around 50 micromole photon per meter per second. The Capapicus will get bleached. Will get bleaching about 2 weeks.
Okay. I think yeah, that's all the questions from the website. Yeah, thank you very much. Maybe we will look later in PA if there's a question in during the discussion. Okay, thank you very much, Dr. Lideman.
You're welcome
for your time. So the next we go to the Dr. Xiao Qin Fu.
Well, me and read already. Well, me.
After this, because after this, and I will return the time to Dr. Nadiarti. Dr. Nadiarti will ask you for the discussion.
Can you hear
so,
me?
Dr. Xiaoting, good
yeah.
afternoon.
Yes,
thank
good
you
afternoon.
for joining us. Yeah, thank you for
thank
joining
you.
us. I have some question for you and also nice video. I will ask also one question, but later if you have still time.
Okay, thank you.
So question, the first questions in here I choose is about the what can you tell for us, yeah, what is the biorefinery principle, and what is the benefit of this method to develop the seaweed production?
Okay, thanks for the question. So my research field is about the seaweed chemistry. Yeah, so we focus on the utilization of the carbohydrates from different kind of seaweeds. So in our topics is about the carbohydrate of carrageenan. For the utilization of carrageenan, we always use the carbohydrates, and the molecular weight is very high, and also the it is always be used as a food ingredient. It's now the industry is to use as a food ingredients. And for our research, we think that if we can degrade the carrageenan to small molecules, which means to produce the oligosaccharide of carrageenan, and its value can be increased. For example, the oligosaccharides have different kind of activities, the different kind of biological activities which can be used in the functional food and also functional cosmetics. That's our research field. So we're trying to develop the enzymes as the tools for degradation of carrageenan. We try to isolate the marine microorganisms and to screening different kind of the carragenase. The enzyme used to degrade carrageenan is called carragenase. So we use this carragenase to produce different kind of oligosaccharide and try to screening their activities and further use it in the high value the industries. So that's why we use the this kind of biorefinery.
So yeah, follow to this question. Does because you use the two enzymes, sulfatase and also carrageenase enzymes.
Yeah.
And does the combination of the both enzymes give the yield of better result?
Okay, very good question. So we have another enzyme related to carrageenase. It is called sulfatase. And this enzyme is used to modify the sulfate groups in carrageenan molecules. And it is mainly used to remove the sulfate groups from the carrageenase. As we know, different kind of carrageenan has different structures. It mainly relies on the position of and the amount of the sulfate group. So we can use the sulfatase to control and to produce the products we want. So I think it's very good opinion to combine the use of these two enzymes. For example, if we just use the sulfatase to deal with the carrageenan molecules usually in the helix or superhelix, the structure. So it is not available for the sulfatase to act on the molecule. So if we first use the carrageenase to degrade it to small part, and the helix structure will be destroyed, and the sulfatase will be more available to act on the sulfate group. Yes, so it's very important. Yeah.
Okay. And yeah, the last questions. Does this enzyme carrageenases have any effect on other carrageenans or specific to certain carrageenans?
Yes.
You explain. You show so many type of the carrageenans, and this is the enzyme only for specific carrageenan, or can be used for all type of the carrageenans.
Yes. And for the carrageenan, it has different structures, and also for the carrageenase also they have different categories. And our research is focused on one very important resource, that is the kappa carrageenan. So so the enzymes we focus on is kappa carrageenase. So if we want to deal with other kind of carrageenan, we should use other carrageenan as the substrate to screening for the very specific carrageenase. For example, the alpha carrageenase or others. So there are other reports also have done. The research also have done carrageenase, but for our research, we focus on the kappa carrageenase. For the isolation and also for the gene cloning work, we get several enzymes. They are all carrageenase. Maybe I think the reason is the carrageenase is very abundant in the seaweed. So maybe also the microorganism focus on using carrageenan. So we successfully purified enzymes.
Okay, it means maybe you can isolate the enzyme from the microorganism or from the other kinds of the Kappa picus or the other kinds of the Euschema, that can be used for the other type of the carrageenan.
Yes.
So I have one question from me, because I look in your presentation that oligosaccharide have an effect also as antiviral. Have an antiviral effect against herpes simplex virus 1. And I just want to know if it is possible for this oligosaccharide from Euschema can be used as antiviral against SARS-CoV-2.
I'm not sure.
Because I look the model or the polar of the inhibition of this oligosaccharide to the virus is inhibit the attachment of the virus on the surface of the cell. It means, maybe it is possible also to use for as anti SARS-CoV-2 because SARS-CoV-2 to also have attachment in the cell of the lung and like that. Is this possible or not?
Yeah,
Yes, I want
think,
to know. Yes, I want to know your opinion. Because
yeah,
in China have many experiment with with the antiviral.
yeah. Yes, actually, in our department, we have several the researchers focused on the oligosaccharides of the seaweed. We are carrying different kinds of activity checking the work. So for the antivirus activity, we also try to screening different kinds of oligosaccharide, not only the carrageenase, but also the from agar and alginate, phycoidin, some other carbohydrates from seaweed. Yeah, and I think it is possible if we can do more the screening work, we can get more useful oligosaccharide. Yeah, because we have the tools. Yeah.
Yeah. We hope. We hope so.
Yes.
Okay. Thank you very much for your time and nice explanation. Yeah. Thank you.
Thank you.
So, I think we move first to Dr. Nadiarti or Dr. Nadiarti. Should I continue or because Prof. Raju, can Prof. Raju waiting for one presentation, one panelist again?
Just continue, Dr. Elmi, to the next speaker, for Ahmad Sadeh.
Yeah, okay. I continue the next. So I invited for the next panelist, Dr. Ahmad Sadeh. Dr. Ahmad Sadeh, can you hear me?
Hello, good afternoon. Dr. Ahmad Sadeh, I'm Prof. Lim.
Oh,
okay.
Okay.
I'm the co-author together with Dr. Ahmad Sadeh.
Yeah, okay. Your name is
Lim Pek Im.
M. Lim?
Yes, correct.
Professor Pike M. Lim. I called you Prof. Lim. It's okay?
Can. Yeah, no problem.
Yeah, okay.
Yeah, because Dr. Ahmad Sadeh have headache after vaccination this morning.
Oh,
okay. So yeah,
doesn't matter. Okay, so yeah, I have a question for you from the website. It is interesting also the video you explain about the interesting initiative by Malaysian government to improve seaweed farming, like using new float, new cultivation method, new kinds of goods, and the production of alternative products like bioplastic and etc. And the question is, how many of these initiative of interesting initiative from the government are being used by the community? I think in the term of the farmer, yeah, community is means the farmers.
Okay.
How many of these? Yeah. How many of these are being used by the community?
Okay, thank you, Madam Chairperson. Okay, so as regard about the innovations that the government of Malaysia have introduced through R&D, so for the floater as well as the cultivation system, yeah, they are practiced by the local community. However, as regards about the harvesting boat, they are actually more suitable for large-scale cultivation. So they are not actually being carried out because at this stage, large contribution of cultivation in Malaysia is actually contributed by the local community, which is a small scale farming. Yeah, thank you.
And about the new innovation, I mean the interesting innovation like new float, using new float, and also using a new cultivation method, is it have been applicable for the community, or still not yet so yeah, so much
Not so, okay.
farmer do
Actually,
that?
for the floats is actually sponsored by the government. So we, for those farmer that's under the government scheme for cultivation, they are all using the floats. So as regards about cultivation system, also they already uptake the method of tying and things like that, because that will minimize the time that is required. Yeah. But for both, it's not being used. Yeah.
And the follow for this question, and for the solution which didn't make it, do you have advice about other innovators to be done?
I think based on our experience, any innovations that is being introduced, I think two main factors that I would like to emphasize. That is low cost, okay, so that is affordable for small scale farmer, as well as also easy to be operated by the local community. So if technology are too sophisticated in terms of maintenance, also will be a problem. Yeah.
So I have a next question. Malaysian government seems to have taken an active role to introduction eucalyptus farming in Malaysia. Can you outline the key steps for the audience, how the government have done?
Okay, so I would say that in summary, the assistance that the government giving to the eucalyptus farming is mainly for the upstream. Okay, so actually through various Malaysia plan, there are different approach have been carried out, but I can summarize that the kind of assistance being given. So one thing is the allocation of the sea area for farming. So the local community, the farmer can access the area without paying any, is free of charge. So as regards for company, they have to apply, but the payment and the fee is very, very minimal. Okay, and then the government also provide the ropes floater for culture, as well as seedling. So, and the government also provide a build a drying platform to be shared by the local community. So it's like just one villages that's formed from different small farmer, so they can jointly have access for the platform, the drying platform. Yeah. So
and yeah, have you finished, or
yeah,
you want
I
to
have finished. Yeah,
more?
no.
Okay.
Okay.
The last question is, what are the key steps in invigorating the industry during the pandemic? Yeah, I think because yeah, many industry was down during the pandemic, and which make eucalyptus industry has declined. Yeah, I mean because yeah, uh, what do you want to government do for reinvigorating the industry?
Okay, I
Again,
think
it means,
yeah, I mean in my talk I share that there's decline as regard about the production of eucalyptus in Malaysia. So, and they are actually due to several factors. So in such as lack of seedling, climate change, instability of price, as well as security. So these are the things that we have to overcome. So the government realized that the importance of that, especially as regards about the overcome the shortage of seedling. So there is a need of R&D to sort for our local seedling, as well as also to identify species that are resilient to climate change, especially for increase of temperature. But however, these are actually not a short-term, not immediate remedy that you can take place. It will take time in order for us to slowly overcome that. Yeah, it will. So I think it will take some time for us to fully actually recover to up to what we are. But that there are efforts that the government are trying their very best. And of course, we need a national seed bank that actually to have a continuous supplies of seed to the farmer. And also a wide supply of seedling will also enable the big company, anchor company that will be able to come in to revise the industry. Yeah.
Okay. I think that's all the question for you. And thank you very much for your time.
Most welcome. Okay.
So,
Thank you.
We go to my colleague, Dr. Nadiati, to continue for the next discussion. Dr. Nadiati.
Thank you, Ibu Dr. Elmi. So now I'm trying to back to Prof. Rajudin Shamsuddin.
Yeah, can you hear?
All right, Prof.
Can you
Your
hear me?
voice now is more
Okay.
clearer. Yeah.
Okay.
Okay. So, Prof. Rajudin, there are some questions, and I'm trying to pick one or two questions from the total question you have. The first question is from Professor Reddy.
Yeah.
Reddy.
Yes.
He said that it is very interesting presentation and hope the outcomes of your study help to generate improved germplasm for commercial cultivation in the near future.
Yes.
So the question is, what are the factors that induce sporulation?
Sporulation. Temperature and salinities is the main factors in
Can you
sporulation.
close to the speakers, to the mic?
Yeah. Two factors that main factors induce the sporulation. That's salinities and temperature. Water. The water. Okay.
Salinity and temperatures.
Yeah.
So, what is the rationale for selecting different colors of light in this study? Because in your video you mentioned about light.
I'm sorry, can you repeat? It's raining here. It's raining here.
What are the factors? No. What is the rationale for selecting different colors of light in this study?
Yeah. Look. The red color is a good color for sports, for release and development. Red color.
Red color.
Red color.
I mean, the rationale. What is the rationale? Latar belakang masalah memilih warna cahaya.
Because red colors influence the reproduction of the algae. Yeah, red colors means is wavelength or quality of the light.
Wavelength.
Yeah, wavelength influence the
related to the wavelength.
Yes, the production of the algae.
What is the best culture medium for growing the spores?
Medium? What do you mean medium? Waters? Because this is be conducted in the water with water medium. I mean. Hello.
Hello.
Yes.
My device is going off. So, what is the best culture medium for growing the spores, Prof?
Medium. I don't understand what do you mean by medium, because we use water.
Culture medium.
Seawater medium.
Okay, so you only use seawater medium.
Seawater medium, yeah.
No other mixed medium or treatment.
No. Seawater.
So until now, only for the seawater medium. Yeah.
Yeah.
Okay. Next question.
Yeah.
Sorry, where is the question? Yeah. Where did you get the fertile parental for the experiment? Was it from the wild or from the cultivated?
From the cultivated. We get
Okay.
the seaweed seed from the cultivated farm. Yeah. That is in Takala Regency of Sulawesi. Yeah.
Next question is, how do you induce the histocaps in natural tally in your lab? Or do you collect thallus during natural histocaps
right.
from the wild?
Yeah, we use thallus vicissitucarp that is get from the farming for the for the farming.
Also not from the
Not
wild,
from the
yeah?
wild. Not from the wild. Yeah.
All right. The square PVC pipe rafts you mentioned in the video with
Yes.
mud bottles are very interesting.
Yeah.
In the rafts, you mentioned both tetraphosphorophytes emerging from these experiments. Can you develop seedlings from both kinds of spores? And do you see any variation between their characteristics?
When we get thallus with spores bearing thallus, we didn't recognize that this is sporophyte or carposporophyte or tetrasporophyte. We just get the thallus with cystocarp, but we didn't recognize that this is as tetrasporophyte and carposporophyte. After seeing the result, that there is spores released in the form of tetraspore and carpospore, and let's realize that actually we use a tetrasporophyte, carposporophyte thallus. Yeah.
Okay. Yes. Do we still have time here? Oh.
Yes.
One more question. Have you ever considered hybridization of different strains of seaweeds using spores to develop better strains?
I think so. I think so. You can do it.
Okay.
Yeah.
Okay, another question. Sorry. You are one of the few groups using spore production as opposed to vegetative propagation. Can you please just go over the reason again? And would you describe this as a best practice?
Yes, as I mentioned in the introduction, that the previous studies have been done by several authors about the spores. That the spores is a good for seedling red algae actually. In terms of the the influence of the environmental factors compared to thallus cutting. Yes.
Okay. All right. Okay, because we are running out of time. Thank you very much, Prof. Rajudin, for your great response and answers. And it's very interesting. And now I will move to the next panelist. Thank you, Prof. Rajudin.
Thank you, Khaled, friends. Yeah.
Okay. So now I invite the next uh, the next panelist. Hello, Professor Delinduan. Are you there? Hello. I saw in the participants list, I saw the name of Prof. Duan.
Yeah.
Can you hear my voice? Okay. Okay. Nice to meet you again in this meeting Zoom.
Yeah.
Okay. Thank you for your time, Prof. Duan. There are some questions here. So as you present in your video about the present status and trends of carrageenan production in China, there is a question from the immense experience China has in seaweed farming. What do you see as important milestones to be crossed for eucalyptus farming?
Well, early in the last 1980s, our pioneer researcher, Professor Wu, conducted the equinoid farming in Hainan Islands, and they actually successfully established the culture system and conducted the large-scale cultivation after 5 years trial. However, after another 5 years trial, because of main causes of feed, a kind of feed like the Incuma very much as a feeder, favorite foods to eat them. So actually, in total, in 10 years, all the cultivation biomass decreased dramatically. So in that time, after now the successful continuous support from the central government, so they do not continuously conduct this work anymore. And in these recent years, from 2000, even though our government started some programs, actually, you know that both the coastal environment and the labor cost are changed greatly. So finally, the large scale conduction of the cultivation of Kuma do not get any actual achievement now. And so far, we are mainly focused on the shipment from Southeast Asia, use these raw materials, and mainly focus on the production of carrageenan and ship out for world markets now. Another is that we are now focused on the bioenzyme extraction measures for carrageenan, different types of carrageenan, or different testing for functional uses in the future now.
Okay. The next question. China is the world's largest cultivator of brown seaweeds in colder waters. However, it seems the introduction of eucalyptus from the Philippines to China is one of the few places where the industry has not expanded significantly. For the benefit of the audience, please could you briefly outline the history, but specifically indicate why you do not think farming of eucalyptus became more successful?
Well, actually, many changes happened or occurs in these past 30 years because of maybe the main locations were now in Hainan province. Maybe every colleague, if you visit Hainan, there are now no ideal place there, I mean, for conducting this type of red audit. And we had to restore our initial programs, even though our government encouraged this type of work. However, compared with the cost, I mean, labor cost, input, and everything, the basic cost for manufacture their cargin is much higher than we shipped from outside of China. So that's why our both central and local government mainly focus on shift the target to the how to process a more fine good cargin in different types of biochemical, chemical, or enzyme measures, and upgrade the products with a high value for future potential applications in the futures. Actually, compare with that, Indonesia, Philippines, Malaysia, the labor cost comparatively now is a middle or lower. And now in China, labor costs reach greatly, and you cannot hire a good fisherman to conduct this type of work now. That's why we had to change this type of strategy and shift to mainly focus on high value of cargin products for medical, biomedical, or other applications in the future.
Okay, so because of the labor cost, it's very high higher in China compared to the Malaysia, Philippines, and Indonesia. Okay.
Indeed, we hope to do joint work with the enterprise or university or research institutions in these countries together, try to do more work independently or jointly, and use your advantages, your materials, your labor cost, and use the works in Indonesia, for example, and back some works back in China. I just write back from a governmental meeting. We will start joint projects on this now. Maybe it will be started at the end of this year.
Yeah, that's a great opportunity to be discussed further. Okay, another more question. What are the major lessons you suggest being important to others trying to make a business out of growing eucalyptus in their coastal waters?
Well, maybe many both Indonesia local citizens and some Chinese which now immigrate to Indonesia to somehow have controlled the market price, to my understanding, or no. Yeah. So that's the problem. The market price from the raw materials to the semi-dried materials, there are very high difference. So if there are good lessons for both of us to establish some agencies, this will be either similar in Philippines or Malaysia. If we control the price on the middle level, I mean, that will be good for both of us to conduct other works on high value purification or extraction in the future.
Okay. All right, then. Thank you very much, Professor Duan, for your excellent, interesting answers. Because of the time, I will move to the next panelist. I invite Dr. Pepijien van Oort. Dr. van Oort.
Yes, I am here. Hello.
Hi, nice to meet you.
Nice
Although
to
only
meet you.
in virtual.
Yes.
Okay. How are you, sir?
I'm fine, thank
Good
you.
afternoon. Is it
Yes.
afternoon in your place now?
Yes, yes. So I'm lucky,
Okay.
much better than the Americans.
Yeah. Okay. Thank you.
How are you in Makassar?
Yeah, in Makassar now is a heavy rain. My background with a heavy rain now. We are in the afternoon. Okay. So, Doctor, how do I call you? Dr. Van Ort. Is that okay? Sorry for
Yes.
my mispronunciation.
No,
Okay.
that is fine. That's fine.
Okay, thank you. In your video, you are talking about the bioeconomic seaweed model, right? For modeling Capapicus cultivation in Indonesia. So the question is, in your model, the growth rate seems to be taken as a constant. The growth rate can vary a lot in the same site depending on the season. How does your model adapt to changing growth rates?
Yes. So I agree. The growth rate can differ quite a bit depending on the environment. And so what we want to do is to estimate the growth rate parameter in 6 sites. And in the same sites, we are measuring the environmental variables. And then we hope to establish how the different growth rates depend on the environmental variables.
Okay, so your model can adapt, yeah, to the changing, to
Yes. Yes,
for the
certainly.
changing.
Yes.
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 Okay. All right. Next question. It is a bit like the chicken and the egg. Which come first, the seaweed farm or the modeling?
Yes. So I like this question. We become a bit philosophical. So first, let me say, why do we do this research? We do this research out of scientific curiosity, and we do this research because we hope to help in the end that this research can help the farmers improve their livelihoods and the whole sector to improve itself. And as you will know, working at the university, it's a never-ending process. You formulate hypotheses, you do an experiment, but then from the outcome of the experiment, you have so many new questions that arise. So you make new hypotheses and you make new experiments. This does not mean there is no progress. In the whole cycle, it is a never-ending cycle in which we do gradually build up the knowledge. And you can see our model in the same way. Actually, our model is a coherent set of hypotheses which we are testing. I hope that answers the question. If not, you can get back to me.
Okay. Yes. So
respond
modeling
immediately.
So modeling just like the other research, never-ending research. There is always a new question, and all of these are to improve our livelihood. Yeah, in terms of seaweed, especially in this case. Okay. Another question. Based on your models, they can be site or country specific. But what do you think will be the next big thing in precision phyconomy based on your modeling experience?
Yeah. So actually there are 2 questions. One is if it is site or country specific.
Yeah.
And the second question is what will be the next big thing. So let me answer to the questions one by one. When we speak of a model, we should distinguish between the equations and the input data. And so it is possible that the equations are the same for every country, but that the input data are different for a particular country or a particular site. So in one site, there may be a different strain of Capapicus cultivated, and this may result in a different growth rate. And of course, if in one site the temperature is different or the nutrient concentration is different, that would lead to different input data to the model, but still it would be the same equations which we are using. And that's the way we are trying to move forward. And we want, yeah. I hope that answers the first question. The second question is, what will be the next big thing? Yes, well, I think we should be modest. There is still a lot we do not know. We sometimes, what I hear from Sulawesi is that in one side, kappa farmers are successfully cultivating kappa ficus, and then 1 kilometer further to the south, it doesn't grow. Well, you would think the environmental conditions are quite similar, and also they try the same strains. So often we don't know. There is quite a bit of experimentation, but not so much in modeling. And also the protocols that are being used are different in every experiment. So I think there is still a lot of things to be gained just in getting the basic science right by using slightly different models, and getting away from this exponential growth towards a sigmoid growth model, and using the same protocols in different experiments so that they can be combined. And also what I find very interesting is to combining the biological and the economic models. What we see in this research is that it's often biologists doing their research with little knowledge of the economics, or it's people in economics looking at the whole value chain with little knowledge of the actual cultivation process. And I think it's interesting to look at these two things together.
Okay, so yeah, so more research. We never stop to find more information and to be help in modeling, and also in economic and ecological sectors. Also, it's very important to be combined, especially in your modeling. Is that something like that?
Yes.
Okay. All right. I think the time is almost finished. And thank you very much for your great answers. Very interesting. And hope we have many collaboration in the future. Thank you. Thank you again, Dr. Van Oort. All right. I think this is the last panelist, and because of the times running out, and we have to say that this is the end of this session. And again, thank you very much for all the excellent panelists, very interesting answers. And let me make a wrap up, a little wrap from our discussion. Because in this session, we are talking mostly about the seaweed from the Southeast Asia, right? So I'm trying to make a little conclusion that actually in general, eucalyptoids has a big promising prospects to be developed for to the big international markets. We have some countries here. There are several countries excellence in production raw materials, and another countries excellence in producing extraction and chemical process to have high values for the seaweed products. So we hope tomorrow we will discuss about the potential collaboration among us. Yeah. So please, all the participants, we still open opportunity for all of you to put your questions in our web forum if you still have any questions. And to all panelists, you can see from the chat room if there is another question that cannot be answered at this time, or and also please go to our web forum to check if there is more question for you all as panelists. Tomorrow we will meet again at the fixed schedule. Please go to the web forum to see the schedule. We start at 2 p.m. tomorrow, Makassar time. Nice to meet you. I'm so happy to be here as a facilitators with all the great panelists. And I hope we can join again tomorrow in other session. That's all from me, and thank you very much again. And I will give this time to the host or committee. Ibu Widya, are you here? Ibu Widya?
Yes, I'm here, Ibu Nadiati.
Okay, Ibu Widya, maybe you can explain what is the agenda for tomorrow, and yeah, and close this session for today.
Yeah, thank you very much, but I think you already closed it. Yeah, and all of the information that you already mentioned, but actually for the next agenda for tomorrow, all of the information we already put it on the website, so you can visit the website and see the next session for tomorrow. That is for session 3 and session 4 for tomorrow. Yeah, I think that's all. Thanks for all of the attendees and all of the great panelists. Yeah, maybe from the TPCD or Alan, do you have something to say before we close?
Well, I just want to add my thanks to everyone who's contributed so freely and with tremendous amounts of information. I think we've heard some challenges, but we've also heard lots of opportunities, and some of the opportunities I see is collaboration amongst you. So I can see people who have the need, and I can see people who have some of the tools to help. So I really do encourage that melding of the minds and forming of groups with the haves and the people who can provide some assistance. So thanks to everybody for so freely sharing the information, and look forward to the same lively interactions tomorrow. Thanks to everybody.
Thanks.
Thank you. Bye-bye. See you tomorrow.
Thank you.
See you.
See you tomorrow. Thanks, everyone.
See you tomorrow.
Bye now.
Bye-bye.
Bye, Bill. I'll see you tomorrow.
All the best.
Thank you. Thank you for
Thank
all
you.
panelists. Thank you so much. Thank you. See you
Oh,
tomorrow.
thank you. See you tomorrow.